Welcome guys. Today we are going to discuss about COVID-19 and CAR T cell therapy, which is chimeric antigen receptor T cell therapy. As you all know that scientists are trying to discover new ways to tackle COVID-19. One of the interesting cell therapy which is very modern is CAR T cell therapy. So let's begin. For CAR T cell therapy, these two gentlemen, Allison and Honzo, got Nobel Prize in Physiology and Medicine in 2018. Basically, their discovery is about a cancer therapy by inhibition of negative immune regulation, which means that there are cancer cells. These cancer cells have a particular antigens. Now we have T cells as our immune cells in our body, which has capability to identify this cancer cell and destroy them by binding to this antigen. But what cancer cell do is they mask this antigen by making more number of antigen around it, and in this way these T cells are unable to recognize this cancer cell, and that's how they mask themselves from the immune defense mechanism. So what is this therapy about? So in this therapy, what they do is they take T cells from the patients and then they insert a new gene, which is CAR gene, chimeric antigen receptor, which makes this T cell rich in these antigenic receptors. And after cultivating the cells in the laboratory, these are reinfused in the patient. So number of T cells which has chimeric antigen receptor which can identify cancer cell are in larger number. So because of this, they can identify cancer cells and destroy them. One more thing which T cells can do is they can recruit more number of immune cells to that particular location, and in this way they can destroy cancer. Now let's see how this strategy can be used for COVID-19. So for COVID-19, the strategy is that T cells are taken out from the patients. Now these T cells can be inserted with SARS-CoV-2 genes. This can be through a virus mediated, or it can be just an mRNA electroporation. After this, these cells, as you can see, are rich in CAR T receptors. The chimeric antigen receptor. These antigen receptors can be identified by these pneumocytes, which are already presenting SARS-CoV protein. So for type 2 pneumocytes, just see my last video in which I discuss about COVID infection and immunopathogenesis. After these receptors are identified by this CAR T receptor, these T cells produce granzymes, perforins, or kind of components which are toxic to these cells. And this way these cells are destroyed along with the viruses. And this way a CAR T cell therapy can be a good strategy to tackle COVID-19. But there is a problem. The problem is identified as this has side effects which are identified in treating cancer patients, these side effects are fever, nausea, and a possible cytokine storm. If you remember from COVID-19 immunopathogenesis, these are the pathogenesis of COVID-19, and hence this may severe down the COVID-19 cases instead of tackling with this. Also, the T cell culture into multiple chimeric receptor take a longer time, which may be from 3 to 4 weeks, which could be even a minimum time. And in this period of time, when this COVID-19 cases are severe, it is really difficult to use this strategy for COVID-19. I hope you all have enjoyed this video. Thanks for watching. Bye bye. Take care.